Very good afternoon to all of you. Today we are going to see the important part that is associated in immunology, that is called as the antigen and its specificity. So let us understand what are antigen, what are its roles that are associated with the immunological systems. So antigens. So what are antigens? We need to concentrate on antigens because this is an important point that is associated with the immunological systems. So let us understand the antigens in a very correct and perfect manner. Substances that are capable of inducing a specific immune response are called as antigens. So this is a very basic definition that is associated with the immunological terms antigens. So these antigens are nothing but they are substances which are able to elicit or start a specific immunological response. They can be recognized by the surface antibodies or by the T cells receptors when associated with MHC molecules. Here, you can see here the viruses or any sort of bacteria have specific sections on their cell membranes or on the outer side of their cell, and these are nothing but the antigens. They can bind specifically with the antibody or TCR. So they again have specificity that is associated with terms of antibody. Now, moving further. Classification of antigens. There are basically two types of antigen that are associated. One is called as exogenous antigens. One are called as endogenous antigens. So what are exogenous? Exogenous antigens are those antigens which enter into the host body from their surrounding or external environment. These are basically pollutants, microorganisms, pollens, drugs, etc. Now, exogenous antigens. What are these exogenous antigens? If you are walking inside a field and if you get something inside your nose, some dust particles, and these you start sneezing, then that is again considered as an antigen to the human body. Means exogenous. Exogenous, what the term means? It is from outside or it is from the external environment by which it is entering inside us and ultimately producing immunological responses. So second type of antigens are called as endogenous antigens. What are endogenous antigens? Xenogenic or heterogenic antigens, allogenic or idiotypic antigens, or autologous antigens. Means ultimately these antigens are produced inside the body itself. They do not come in contact with the external environment, and ultimately these are considered as endogenous antigen. Endo, inside, exo, outside. Correct? Fine. Moving further. Now, what are xenogenics? Again, in terms of endogenous antigens, there are antigens called as xenogenic antigens, foreign items which are related with tissue transplantation or serology. Means ultimately, if a particular person has undergone a serology or what we can say transplantation, then ultimately that tissue which is inserted from the external part inside the human body may act as an antigen. So therefore, it is considered as a xenogenic antigen. Allogenic antigens. So foreign items which enter when an individual receives a blood transfusion or undergoes a transplantation operation, then these are called as allogenic antigens. Autologous antigens. This group of antigen is very rare and unnatural. In normal conditions, self components are non-immunogenic in nature, but in an abnormal condition, self-body components are started to consider as non-self and antigenic component. Means ultimately in terms of autologous antigen, what happens? The body itself start to mount a reaction onto cells of its own body, and therefore it is called as autologous kind of antigens. Now, what is immunogenicity? Ability to induce a humoral or cell-mediated immune response is called as immunogenicity. So, antigenicity, ability to combine specifically the final product of humoral or cell immune response is called as antigenicity. All molecules possesses the property of immunogenicity, all possess the property of antigenicity. Some molecules that possesses the property of antigenicity but lack immunogenicity are known as haptens. Means immunogenicity is what? Immunogenicity is the ability of that particular antigen to produce or to start an immunological response, either a humoral or cell-mediated immunological response. And antigenicity, it specifically binds to the product of the immunological response, maybe humoral cell-mediated immunity. So once the immunological response gets started and the formation of certain types of antibodies or cells from the body starts, and when the antigen binds to that cells, only then we can say that antigenicity has been produced. You can see amino acids, haptens, lipids, so on and so forth, carbohydrate and proteins are said to be highly antigenic in their nature. So haptens are molecules which actually start the immunological response but do not specifically bind with the product of the immunological response. Correct? Immunogenicity, contribution to immunogens. Now, what are the properties of immunogenicity? Why an antigen is said to be an immunogen? It should be foreignness, molecular size, chemical composition, physical form, degradability. See here. First is foreignness. The immune system normally discriminates between self and non-self such that only foreign molecules are immunogenic. When an antigen is introduced into an organism, the degree of immunogenicity depends upon degree of foreignness. Means anything that enters inside the body which is foreign, that is said to be a immunogen. Second property is molecular size. This is not absolutely size above with the substance while immunogenic. However, in general, the larger the molecule, the more immunogenicity it has. The best immunogen that tends to have a molecular mass approaching 1 lakh Dalton. The poor immunogens have a molecular size of 5,000 to 10,000 Dalton. Means ultimately, for an antigen to be an immunogen, it has to be, its molecular size has to be at least 1 lakh Dalton, and then and only then it is able to contribute itself in starting an immunological response. See here, glucose molecules, very small, angiotensin. See here, there is a molecular weight that has been given. Hemoglobin, fibrinogen, 4 lakh, via globulin, 1 lakh 50,000. The antigenicity has increased here. You can easily see here the examples that have been given. So antigenicity ultimately increases with the molecular size. Chemical composition. In general, the more complex the substance is chemically, the more immunogenic it will be. The addition of aromatic amino acid like tyrosine and phenylalanine to synthetic polymers can have a profound effect on immunogenicity. All 4 levels of proteins can contribute to antigenic determinants and hence they affect immunogenicity. So the complexity of that particular chemical is said to be one of the important part. The more chemical composition the protein molecule would have or that antigen would have, then it is said to be highly immunogenic. Physical form. In general, particulate antigens are more immunogenic than soluble ones, and denaturing antigens are more immunogenic than the native form of antigens. Means ultimately, the smaller the physical form of particular antigen, it will escape the immunological systems or the cells will not recognize it, and ultimately it will cause an more antigenic effect on top of it. Moving further. Degradability. Antigens that are easily phagocytized are generally more immunogenic. This is because the most antigens develop an immune response requires the antigen to be phagocytized, processed and presented to the T cells or the APCs or antigen presenting cells. Means ultimately it has to be degraded by the immunological system. For example, the phagocytic cells are there which are helpful for degrading this particular molecule. So once these molecules are degraded, then and only then it will be recognized by the immunological system, and ultimately the immunological system will start an immunological response. Now, are there any other factors which are associated for this particular kind of thing? So what are these factors? Genetic factors. Some substances are immunogenic in one species but not in another. Yes, of course, genetic factors are said to be important part. Similarly, some substances are immunogenic in one individual but not in others. The species and individuals may lack or have altered genes that code the receptors for antigens or B cells and T cells. Means ultimately, for example, if we go along with, if you are genetic makeup is said to be strong, you will not absolutely suffer from any sort of infections. For example, if we are now the winter season is coming, so maximum of us will certainly go with infections of the respiratory tract. But if genetical factors are important, wherein your body cells, if they are being strongly been made due to your genetics, then certainly you will not have the drastic effect of the cold weather. Next point that comes in place, age. Age can also influence immunogenicity. Usually very young and very old have a diminished ability to mount an immunological response to an immunogen. So when we adults are there, our immunological systems are very very correct or very up to the optimum level. But when the child is there, it has a maximum danger of being getting infected by various different diseases. While old people also, their level of immunogenicity has decreased, and therefore they are prone to have the infection in a very larger amount. So this is all for today. What we have seen in terms of antigens, the basic part as well as the factors that are essential for antigenicity.